One of the first things we did was try and see, is there any kind of genetic susceptibility to infection and to control of infection? So we had this phenomenal setup where we were able to recruit hundreds of patients. We could determine viral load on each of them, how much virus is in the blood, and then we could do these genetic measurements of something called the HLA molecule. HLA molecules basically present viral peptides in an infected cell to the immune system by taking them and putting them on the cell surface. And when we did this study, what we found was that HLA-B alleles were strongly associated with control of infection. So here was really the first evidence that there were genetic differences, not racial differences, but genetic differences in terms of HLA, in terms of susceptibility or resistance to disease progression. So the next question was, is there a relationship between viral load and what's targeted? In other words, that little blue thing that's sitting in the HLA class 1 binding groove, which is the viral peptide, does it matter where that came from, what part of the virus? So we again took a very large population study, looked at relative targeting of the different viral proteins, and then determined if that caused better or worse outcome. Well, what about these cytotoxic T cells? Well, here we looked at something that had just been described in a mouse model, something called PD-1, and some of you may have heard of this. It's a negative immunoregulatory molecule. Basically what it does is it gets upregulated on these killer cells and tells them to shut down. And the question was, could it be that in fact these cells are shutting down prematurely? And in fact, that's exactly what we found when we looked at this, was that when we looked at the HIV-infected patients on the left, they had this very high upregulation of PD-1. And that was leading to dysfunction of the T cells. So now we have three different important clues that tell us what's modulating control of virus infection in these individuals.